Alright guys, so sorry about the confusion yesterday. This is going to be the third time that I've covered the beginning of this particular PowerPoint. Had some issues with my laptop at home, so this lesson is going to cover the whole lesson of protein synthesis, but in class it's going to be broken up into a couple of sections. And so this is actually our next to last lesson for the semester. You guys are most likely just coming back from Thanksgiving break, and so I hope you guys had a wonderful Thanksgiving. And we just have a little bit more to go before the semester is coming to a close. And so we have the lesson on protein synthesis as well as a lesson on gene regulation. Now, something that's a little bit different is that we are not going to have a test over this particular unit. So far we've covered DNA, its structure and history and replication. And then we're going to have the protein synthesis and gene regulation. But instead of having a test, you're going to have some questions on your semester exam. So we will have some quizzes, and we will have a lab quiz and some activities, but we're not going to have an actual test over this unit. So just to kind of start off protein synthesis, in order to understand it, we do have to understand a little bit more about the RNA molecule. And so back when we covered macromolecules, you learned about DNA and RNA and at least some of the basics about it. But when we're talking about RNA, there are three main types. We will talk about a couple of others a little bit later in the lesson. And so we have messenger RNA, just kind of abbreviated as mRNA. And this is going to be one of our major players in protein synthesis. We have ribosomal RNA, or rRNA. And this one is pretty much only going to be mentioned here. It is part of the structure of the ribosome. So back when we did organelles, we learned that ribosomes are pretty much composed of RNA and protein. And this is the type of RNA that you will find as part of the ribosome structure. I'm sure it has a more specific role in protein synthesis, but this is all that we need to know for this unit. Now the third type is transfer RNA, abbreviated tRNA, and like its name implies, it's going to be transferring something. It's specifically going to be amino acids. And this is going to be one of our major players in translation. So there are three main differences between the structure of RNA and the structure of DNA. They are both polymers of nucleotides. However, one main difference is the sugar. Where DNA had deoxyribose, RNA has ribose. They're very similar in structure. They're both 5-carbon sugars, but ribose has an oxygen that deoxyribose does not, hence the name deoxy. There's a phosphate group in all of the nucleotides, just like we saw with DNA. The sugars and the phosphates make up the backbone. And then there's going to be 4 different nitrogen bases that you can find in RNA, and this is our second major difference, and that there are no T's in RNA. Instead, they have a base known as uracil, which is similar to thymine. It's going to be a pyrimidine, just like thymine was. Thymine would bind to adenine, and the same is going to be true of uracil. So pretty much any place where you would have found a T in DNA, you're now going to find a U in the RNA. And so when it comes to your complementary base pairs, A will pair with U now instead of T. A T on the DNA would still pair with an A on the RNA. And then the third main difference is that RNA is typically single-stranded. Now it can fold up to take on unique shapes, but it's typically just one strand of nucleotides, whereas DNA had 2. And so this whole lesson is all about making proteins, and so I do want to go back and just do a little bit of a review about what proteins are. They're another one of our 4 macromolecule groups. And so they are polymers made up of amino acids, and there's 20 different amino acids that exist. Some of these we can make, and some of these we have to consume in our diet. Each of these 20 different amino acids are different because of their R groups, which gives them unique chemical properties. And so a basic protein is an amino acid chain linked by peptide bonds, and this is why we call it a polypeptide. And so going back to our macromolecule lesson, there are 4 different levels of structure in proteins. And so the basic structure of a protein, or what we call the primary structure, is the polypeptide, which is just a chain of amino acids, okay? So amino acid linked to amino acid linked to amino acid, and these are my peptide bonds in between them. A typical protein is probably going to be about 100 at least amino acids, maybe less, but probably more. Now, this right here is not a functional protein, but when we're talking about protein synthesis, this right here is what we are making, the basic polypeptide. Now, because of the interactions of R groups and chemical properties, this polypeptide can go on to twist or fold like an alpha helix or a beta pleated sheet. We've got more bonding involved, like hydrogen bonds. And then we have a more tightly, very specific type of folding that will lead to the tertiary structure. And this is what we call the globular structure. It has a very specific shape, which gives it a very specific function. And so it's starting in the tertiary structure that we have a functional protein. And then the way you reach quaternary structure is if you had multiple polypeptides. So like for instance, hemoglobin has 4 different polypeptide subunits. And so you would essentially have 4 different polypeptides in that final structure. And so these are my functional proteins here. So what we're building in protein synthesis is this basic amino acid chain called a polypeptide. Now, how do we do that? It's determined by our genes in our DNA. And so we kind of start learning early on in science that DNA is our genetic code, or DNA makes us who we are. Well, the reason it makes us who we are is because it codes for the proteins that we make, and it's those proteins that actually give us our traits. So pretty much you have segments of your DNA known as genes, and those genes are going to code for a particular protein. So when we're looking at the process of protein synthesis as a whole, we can actually summarize it in a very clear, concise statement. Okay, this little statement right here is what we call the central dogma of biology. It's very essential to how life works. And so we have DNA, which has the original code. Now, the thing is, DNA stays in the nucleus. Okay, it cannot leave. It is double-stranded. It is too big to fit through the nuclear pores. And so the DNA is going to stay in the nucleus. Now, the problem is that the proteins are made at the ribosome. So the instructions for the proteins are in the nucleus, in the DNA, but we're going to be building those proteins at the ribosome. And so we kind of need a go-between, and that is going to be the job of RNA. So this is why we have the overall statement of DNA to RNA to protein. Now, we can actually break this down further, and in this one little section of our notes, we have some very important information. DNA to RNA, just the first half, okay, that is going to be transcription. And so transcription is the process of making the RNA from the DNA instructions. Now, if you ever get confused as to which one comes first and which ones comes second, they're in alphabetical order. Transcription with the C comes first, translation with an L comes second. So transcription is the first part of the statement, DNA to RNA. Now, technically all RNA is made by transcription, but I do want you to start getting in the mindset and start thinking about mRNA when we're talking about this little statement here. So we're going to make our mRNA our messenger, and then we're going to use that messenger RNA to build the protein. And that is the process of translation. Okay, so DNA to RNA is transcription, and because that involves DNA, that's going to happen in the nucleus. And then building the protein from the mRNA instruction is translation, and that is going to take place at the ribosome. So there's a lot of very important information in this one little segment right here. So we're going to kind of take this and expand on it a little further and go into the details of these processes. So this picture is just kind of showing you the overall process. So we're going to start in the nucleus, okay, and you can see that here's my nuclear envelope. This is a large nucleus here. We're going to start by taking the DNA and turning it into RNA. That's the process of transcription. We are going to process that RNA a little bit, make some adjustments and some edits to it. Then the RNA is going to leave the nucleus. It's going to pass through the nuclear pores. It's going to be attaching to a ribosome somewhere out in the other parts of the cell, and that is where we're going to build our proteins in the process of translation. And so the final product is going to be this basic polypeptide. And so we're going to start with the first process of transcription. Once again, that is DNA to RNA. And as I mentioned earlier, all RNA is made in the process of transcription, but I do want you to kind of start thinking about, for our basic protein synthesis flow, we're going to be making mRNA, okay, our messenger. And so remember that DNA is in the nucleus and it cannot leave. And so this process is going to look kind of similar to DNA replication with some key differences. One is going to be the enzyme that is catalyzing this reaction. So back when we did replication, we had DNA polymerase, and the job of DNA polymerase was to build the DNA strands by adding the complementary nucleotides. If you remember, DNA polymerase could only add and build the strand in one direction. It had to have a primer to build off of. RNA polymerase is going to be picky, but it's not quite as picky as DNA polymerase. And so the enzyme RNA polymerase is going to be responsible for building our RNA molecules. And so there's going to be three main stages to transcription. We have initiation, elongation, and termination. And these are actually really pretty simple. So initiation starts when the RNA polymerase binds to a promoter. Okay. Now, just to kind of explain a little bit more about a promoter. So not all of our DNA is protein coding. In fact, only a small percentage of our DNA is actually code for proteins. A lot of the other DNA, which we used to just refer to as junk, but there are definitely some functions of the other portions. A lot of our other pieces of DNA are regulatory sequences. And that's essentially what a promoter is. It typically has the sequence TA, TA in it. But what the promoter is it just signifies the beginning of a gene. So it is just a sequence of DNA nucleotides, but it's kind of essentially saying, hey, start here. One of the differences between replication and transcription is that in replication, we copied the whole DNA molecule from beginning to end. And we also copied both sides of the DNA. Whereas in transcription, we're only going to be copying one side of the DNA, and we're only copying one particular gene. So the RNA polymerase has to know where to start, and that's going to be our promoter. So the RNA polymerase binds to the promoter. In elongation, that RNA polymerase is going to move along the DNA sequence, and based on the bases and the complementary system, it's going to build a single strand of mRNA. And so remember that when we're talking about transcription, if there was an A in DNA, it would now be paired with a U. Now, if there was a T in the DNA, it's still going to go with A. Okay, so remember that U is just replacing thymine in the RNA. And then your C and your G is going to stay the same. C goes to G and G goes to C. There's nothing different there. Okay. So RNA polymerase is going to build the mRNA strand by adding new nucleotides. Just like we saw in replication, it's going to build the new strand from the 5' end to the 3' end. And then, once again, we're not copying the whole sequence. So how does it know where to stop? And so in termination, the RNA polymerase reaches another sequence called a terminator, which just signifies the end of the gene. So it's really pretty simple. The RNA polymerase binds to a promoter. It builds the new RNA strand based on the complement system, and then it stops when it reaches a terminator sequence. And so it definitely looks a little different from replication. Notice there's only one strand here. They're using a different color to signify a different sugar. Remember, there's ribose in this strand. And then notice there are no T's. Any place where there would have been a T, there is now a U. So as I mentioned earlier, all RNAs are produced by transcription. Now the one that we really care about is the mRNA. In terms of the process DNA to RNA to protein, we're making mRNA in that transcription stage. But our tRNAs are made this way, our rRNA is made this way. Here's a couple of other types, these small nuclear RNAs. We're going to see those and what they do here in a moment. There's microRNAs, which are just little tiny RNA sequences. And so there's going to be some other functions of RNA. Remember that tRNA transfers, ribosomal RNA makes up the ribosome. These guys help to edit the mRNA. MicroRNAs can serve as catalysts, and so there's a variety of functions that you can find for RNA. But the two main ones that we're going to talk about are mRNA, and that's the one that you need to remember is being made in transcription in terms of protein synthesis. And then we're also going to talk about tRNA, but they have already been made. Okay, we're never going to talk about making tRNA. So what we made in transcription is what we call the primary transcript, or sometimes you'll see it referred to as the pre-mRNA. So the cytoplasm is a dangerous world, and so before that mRNA it leaves the nucleus, it's going to have some modifications. So on one end, on the 5' end, it's going to get a 5' cap, which if you look down here, it's essentially a nucleotide that has guanine and extra phosphates. Okay, so normally a normal nucleotide just has 1 phosphate. This one is going to have 3. So this is what I call my 5' cap. And what it does is it actually kind of protects the mRNA from degradation because there's enzymes in the cytoplasm that could break it down. But it also tells that RNA where to attach to the ribosome. And so that 5' end is going to be where the mRNA attaches. On the other end, on the 3' end, there's a series of adenine nucleotides, and that's going to help that mRNA get out of the nucleus and to get where it needs to go to a ribosome. Now, in addition to actually putting end caps, we're also going to edit the actual genetic code of the mRNA. Because once again, not all DNA codes for protein. And so within that gene region, there are going to be pieces that do code and there's going to be pieces that do not. It's not like it's just a solid coding sequence. Once again, this whole concept of non-coding DNA is still being investigated. So we don't fully understand all of it. But we do know that there are pieces that code and there are pieces that don't. So the pieces that do not, we call introns. And I want you to remember the phrase junk in the trunk. Okay, the junk are the introns. These are the pieces that are non-coding. And then the exons are the pieces that do code. Okay, so you can kind of think about this like when you're making, or like from the teacher standpoint, when I'm making a set of notes. In front of you have a set of notes, some note pages that are maybe 3 to 4 pages, okay, maybe front, front, or front, back. Now, if you were to go to a college biology textbook and look up the chapter for protein synthesis, you're probably going to find that it's much longer than 3 or 4 pages. And that's because there's extra stuff in the textbook. There's going to be fluff, there's going to be pictures, there's going to be some intro paragraphs and closing paragraphs and summaries. And we don't necessarily need everything that is in the chapter. And we don't need everything that is copied into mRNA. And so essentially what I do as the teacher, as I go through the text in the chapter, I pull out the important stuff. In this case, this would be what we would consider the exons, the things that I want you to know. And I'm going to cut out and just not worry about the extra stuff, the fluff. And so this is what we're doing to our mRNA molecule. We're cutting out the junk, the introns, and we're just putting together the pieces that are absolutely necessary so that we kind of have a final transcript that is only the information that we need. And so before that mRNA leaves, it gets those 2 protective end caps and it gets the introns cut out and put back together. So here we have the whole DNA sequence that was copied. Everything is copied when the mRNA is made, but the introns are going to be removed and only the exons will remain. And so our final transcript is just the protein coding information. Now this is going to be accomplished by these little complexes called spliceosomes. It's kind of a fun, nerdy biology word. And these spliceosomes consist of SNRPs, which are some of those small little RNA sequences, along with some additional proteins. How they do it exactly, I do not know, but essentially they go in, they cut out the sequences, those introns that we don't need, and then they also splice together the exons so that we have that final mRNA transcript. And so everything is always specific, and I'm sure it's based on base pairing, and a lot of it goes back to gene regulation and proteins. Proteins are always specific in what they do. All right, so now this takes us to our genetic code. So at this point, we are almost ready to build our protein, but in order to build our protein, we need to understand how the code works. Okay, so the instructions for building our proteins is going to be in the nitrogen bases, the A's, the U's, the C's, and the G's. Now it's kind of like our alphabet. You know, we can say the alphabet A, B, C, D, E, F, G, all the way through Z. But if all I did was walk up to you and sing the alphabet, I'm not really telling you anything. So how do we make the alphabet make sense? Well, we group letters together. So if I put CAT together, that now has meaning, okay? And more than likely, you're probably picturing a cat. If I put DOG together, you're now picturing something different. So different combination of letters equals different meaning. And that's kind of the way our genetic code works. So we read the code 3 letters at a time, and each 3-letter sequence is called a codon. Now, the codons code for amino acids. So I do want to kind of explain that you have 4 letters in 3-letter combinations. That's 64 possible codons. So these are going to code for the 20 amino acids that exist. So that should tell you that it's not a 1-to-1 ratio. Okay, there can be multiple codons for the same amino acid. There's also 3 codons that we call stop codons. And so they don't actually code for an amino acid. They essentially say this is the end of the protein, okay? Now the start codon actually does code for an amino acid, but the start codon, which is the beginning of our protein, is always going to be AUG, which if you think about us starting school in August, that's usually pretty easy to remember. But this one actually does start the protein, and there is an amino acid that is coded for by that. So the way we read our genetic code is with the help of a codon chart. And so you should have one of these. There's actually a couple of different styles. So you have the wheel, or you have the kind of rectangular chart. But in both of them, you just know how to read them, because you never know what you're going to get on a test. Okay, so I want you to know how to use both. Now, if you have the rectangular chart, and you may have one in front of you, you should have gotten one that's your own copy. But the first letter in the codon is always on the left, and so that will narrow it down to a particular row. Okay, so the first letter will narrow it down to one row. All right, the top is the second letter in the codon. And so what that does is that allows you to narrow it down to a single box, okay? And so like this would mean if it was UU, I'm now looking in just this box. If it was UA, then I'm now going to be looking at this box. Or if it was GG, that narrows it down to this box. We're reading it like a grid. Now the third letter is going to be over here on the right-hand side, and this is going to tell me the specific position within that box. So you can see if I had U, 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 what I would do is I would come all the way across, and that tells me that that is phenylalanine. So the first letter tells me across, the second letter tells me what box I'm in, and the third letter is going to tell me what position within that box it is. Now the one that you have doesn't have these combinations there. You're going to have to put it together, okay? So that's how you read the first codon chart. Now notice that some of these, like for instance, all 4 of these codons are all coding for leucine, okay? Here's our start codon, AUG. So I go A, U, and then G, and this is going to be methionine. So my start, it actually is an amino acid. It's the amino acid methionine. We also have 3 stops. We have a UGA, we have a UAA, and a UAG. And so that does not code for an amino acid. It just signifies the end of our protein. Now the wheel is going to show us the same thing. Okay, these are going to be my first letter, the biggest circle on the inside. The next is going to be my second letter, and the outside ring represents my third letter. So to use the wheel, you just go inside out. So here's A, U, G, and there's my methionine, okay? If I looked up UAA, there's my stop. CCC, that would be proline, okay? So I just follow the wheel from inside out, and it has the exact same information that you found on the other codon chart. Once again, know how to read both, because on a test you never know which one you're going to get. All right, so the genetic code, once again, read 3 letters at a time. Each 3-letter sequence is a codon, and that codon is going to code for either an amino acid or a stop. Now this code does not just work like that for us. This code is pretty much universal. We would use that codon chart if we were trying to analyze bacterial DNA. We would use that codon chart if we were looking at plant DNA. The basic code and how it works is universal. Okay, so this does help provide evidence of common ancestry and the concept that all things evolve from, ancient ancestors. All right, so now we are going to our last stage of protein synthesis, and that is going to be translation. Okay, this is where we're taking our RNA and building our protein. Now, I do want to remind you that for the sake of that little phrase, we're taking the mRNA, the messenger, and we're going to read it to build our protein. Okay, so the mRNA is going to leave the nucleus. At this point, it's already been edited and spliced to only have coding DNA. And then it's going to attach to a ribosome at that 5' end. And then the ribosome is going to move along that mRNA sequence and read it 3 letters at a time. So remember, we read the code in 3-letter sequences called codons. Now this is where we're going to see the job of tRNA. Now, for the sake of our statement, DNA to RNA to protein, never once have we talked about making tRNA. Okay, these tRNAs are already made, they're already out in the cytoplasm, and they're already attached to amino acids. So tRNA is going to serve as a delivery boy, and it's going to bring the amino acids to the ribosome so that we can build our protein. So I mentioned earlier that RNA is single-stranded, and tRNA is also single-stranded. It just kind of folds up and takes on this unique little T shape or clover shape. So there's 2 important locations on my tRNA molecule. One is at the 3' end. This right here is where a particular amino acid is going to attach to my tRNA molecule. Okay. The other important site is down here at the opposite end, okay, at this bottom loop. Down here we're going to have a 3-letter sequence called an anticodon. So I want to remind you, the mRNA has codons, and you're using a codon chart to determine what amino acids go in the sequence. Please understand, it is always the codon that determines the amino acid. So anytime that you are using your codon chart, you need to be sure that you're looking at the letters on the mRNA molecule only. Do not look at the DNA because it has T's. Do not look at the tRNA, okay? If you're ever using the codon chart, the only molecule that you should be using is the mRNA. But what this anticodon is it's kind of like a check mechanism, all right? So the anticodon will be complementary to the codon. So if I had an mRNA codon that was AAA, then the tRNA that's going to match up with that should have a UUU, okay? So all the anticodon is just kind of a check mechanism. Now earlier we said that there were 64 different codons, but if you notice here, there's only 45 different anticodons. On the tRNA, there's actually another base called inosine, and so it can actually bond with a U, a C, or an A. And so what happens is we don't really have a 1-to-1 ratio. Okay, there are more codons than there are anticodons, and we call this wobble. Now, normally it's in that 3rd position. Okay, so if we go back to our codon chart, I just want to show you that typically, if the first 2 letters are the same, a lot of times it may be the same amino acid. So here, Cu, 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 all of these are leucine. Notice it's that 3rd letter position that may give us a different amino acid, okay? So like up here, if we change that 3rd letter, that changes it from phenylalanine to leucine, all right? So I just wanted you to be aware that that 3rd position is normally where you would find that inosine, and that's where we get that kind of a little more flexible matchup. So the tRNAs are responsible for delivering those amino acids. But this amino acid is always going to be determined by that mRNA codon. Okay, so I don't want you to get that confused. Now what's going to happen is that ribosome is going to move along that mRNA sequence, reading it 3 letters at a time. Now on our ribosome, there's 3 important sites. Okay, really there's 2. There's the A site and the P site. Okay. So the A site is where the mRNA is coming in, and this is where we're going to kind of figure out what amino acid needs to be brought, okay? This is where you will have your incoming amino acid. Now what's going to happen is once that amino acid is brought by tRNA, the ribosome is going to move over. And so that tRNA that was in the A site is now going to be moved over to the P site. It always goes in the order ape. Okay, so that should help you remember that. Now the P site is always going to have the growing polypeptide. And so what will happen is we're going to see this constant shift. When it shifts over to the next codon, the P site will go to the E site, which is essentially where the tRNA is released. Okay, so incoming amino acid, growing polypeptide, your exit site. All right, so we're going to take a look at this in more detail. You have an activity where you have to draw a ribosome. I just kind of want to show you that this is kind of what it's asking for right here. Notice you have a larger subunit, a smaller subunit, and then you have these three sites, the A, the P, the E. So this is where the mRNA will be coming in. Then the A site will shift over to the P site. That one's going to hold the growing polypeptide, and then it's going to shift over to the E site where the tRNA is going to exit. Now there's some videos that can help you. I'm going to show you a visual here in just a moment to kind of help you see it. But the basic process is similar to transcription in that once again we have initiation, we have elongation, and we have termination. Okay, so in initiation, this is where we start the reading of the mRNA. And so it should start with that start codon, which is going to signify the tRNA to bring methionine. Okay, so just to kind of help this make a little sense, I'm actually going to switch over to a pen. So if our DNA was TAC, okay, in the process of transcription, we would make our mRNA, and that would read AUG, okay. Now, when you look up AUG, that is where you get methionine, okay? And so here's my amino acid, and this would be my amino acid methionine. All right? A lot of times you can abbreviate your amino acids with the first 3 or 4 letters. So the AUG is what determines methionine. Now, the tRNA that carries methionine is going to have an anticodon on the other end. Okay, so here's my methionine. The anticodon is going to be complementary to my mRNA. So A would go to U would go to A, G would go to C. Okay, so it's the codon that determines the amino acid. The tRNA has the anticodon just for a match mechanism, just to make sure the right one is being brought to the right place. Now, let's say that the next DNA sequence was GGG. That would be CCC on our mRNA, okay? Now, if you look up on your codon chart, that should be, if I remember correctly, that should be proline, okay? So the tRNA that would bring proline would have the anticodon G, G, G. So remember, the anticodon is just complementary. Now, what I want you to see is that DNA was one long strand, okay? mRNA was going to be that one strand. But notice that our tRNAs are separate individual tRNA molecules, okay? It's not like I have one long strand of tRNA. And once again, we're not making these. These tRNAs are already made. They're already in the cytoplasm. They're already charged, which means they're attached to these amino acids, okay? So all we're seeing in translation is the reading of the mRNA, 3 letters at a time. The tRNAs are bringing the appropriate amino acids to the ribosome, and we're going to be forming peptide bonds between these, okay? So this would come into my A site. I would have methionine. It's going to shift over to the P site, and then my new amino acid is going to be brought into the A. Now, once there are 2 amino acids there, a peptide bond is going to be formed, all right? Then these are going to shift this tRNA is going to leave, and that growing polypeptide will always remain in that P site. Okay, it's just going to kind of constantly keep shifting, keep shifting, keep shifting. We're going to do an activity that hopefully will help you kind of see this a little bit better. The videos may also help you see it a little bit better. And so in initiation, we start with methionine. In elongation, we're going to move along that mRNA, and the amino acids are going to be added one by one, constantly shifting from the A to the P to the E site. Okay. Now, the process will end when a stop codon is reached, and that signifies the end of the protein. So mRNA is being read 3 letters at a time. It starts with the start codon and the amino acid methionine. Each amino acid is being added based on that 3-letter sequence. The codon determines the amino acid. The anticodon is just helping form a match mechanism. And then it stops when we reach a stop codon. So here's kind of a picture that hopefully maybe helps explain it a little bit. Okay, so here's my A site. Okay, my P site, my E site. So initially my mRNA is coming in here with the 5' end. Okay, we're moving along. So this tRNA, once we have 2 in the A and the P, a bond is going to be formed. The A shifts to P, the P shifts to E, and notice this is where my tRNA is leaving. Okay, it can go out into the cytoplasm, pick up another amino acid. So my incoming is always coming into the A site, and we're always exiting at the E site. And it just keeps shifting and shifting and shifting until that stop codon is reached. So here's something else to understand is you can actually have translation happening way more than just one mRNA, one ribosome building one polypeptide at a time. Many ribosomes can read that same mRNA, which is going to mean more proteins, okay? And so you can have that one mRNA read over and over and over again to produce more and more protein. Okay, it's not like it's read once and then it's broken down. So the polypeptide, once it's built, remember that is not a functional protein. So it is going to go on and it will start to interact and change and take on that tertiary structure or that proper conformational shape to become a functional protein. And then there can be either further modification, like in the Golgi, things like that, where they can be targeted, they can be tagged, they can be shipped to different parts of the cell. Now the last thing we're going to talk about are mutations, and mutations are just going to be changes to our genetic sequence. The mutation could occur on the DNA level, the mutation could occur on the RNA level. Now we've already talked about chromosomal mutations, and that's where you have whole pieces or whole chromosomes that can be affected. That would affect many traits because many genes could be part of those chromosome pieces. Now, this lesson is going to focus on point mutations, which is pretty much just little individual nucleotide changes. We also call these SNPs. SNPs. Okay, these are called single nucleotide polymorphisms, which just means single letter changes. Okay, now there's 2 main types. We have a substitution. And this is where one base is substituted for another. Maybe instead of an A C is placed, or instead of a G, they put an A or something like that. Now, it all depends on where that change occurs, okay? Because sometimes it can have an effect, sometimes it can't, okay? Now, at most, a substitution would affect one amino acid. Now, that could still be a very drastic change to the protein. So it can be neutral or silent, or it could also be very extreme. It just depends on where in the DNA or where in the RNA this change happens. Now, it could be neutral, okay? Like, for instance, if I had CCC, if it got changed to CCA, okay, if I look up this mRNA on my codon chart, these are both the same amino acid. So if the change led to something like this, it may not have an effect at all, okay? Now, sometimes it could be what we call a missense, where it just changes the amino acid. Maybe instead of proline, it caused it to code for an amino acid like leucine or something like that. Now, a nonsense is where it actually changes an amino acid to a stop. So it causes a premature ending to your polypeptide. So once again, it all just depends on where, okay? But a substitution at most will change one amino acid, but it may not change it at all. It all depends on where it happens. Now, the other type is either a deletion or an insertion. If you add nucleotides or subtract nucleotides from the sequence, remember the code gets read in groups of 3. It doesn't matter. If you have a mutation, the ribosome's not going to recognize that there's a missing letter. It's just going to keep reading in groups of 3s. And so what will happen is it will shift the reading of the 3-letter codons. And we call this a frame shift. So pretty much just about any amino acid after a deletion or an insertion could lead to a completely different protein. Now, any mutation could be bad. Any mutation could be good. It all depends on where and what. But definitely a deletion or an insertion or a frame shift is going to lead to a greater change in the protein. This may affect one amino acid, could still be bad, but this is going to affect many amino acids. Now, anything that could lead to a mutation is called a mutagen, okay? So like things like chemicals, things like radiation, toxins, anything like that could be considered a mutagen. And so most of our mutations are going to be these types, okay? We can accumulate these. Remember, DNA replication isn't a perfect process. Transcription is not a perfect process. There could be changes in any of the areas where we're copying genetic code, okay? So here's an example of what it would look like. So here's my original. Here we have a substitution, and it changed an A or a T to an A in the DNA, which led the change to a U here. Notice everything is the same except for this one amino acid. Now, that could still be problematic because that protein's not going to fold the exact way that it should, possibly. Okay, so it may or may not have a great effect. It depends on what the amino acid is, what the chemical properties are. But you can see here that we only had one amino acid changed. Now, down here, we had a frame shift. Okay, so GGG or TAC, AUG, GGG, CCC. What happened is a letter was taken out or added here. Okay, so let's see. I'm trying to find where the mutation is. TAC. It looks like it can't really see the mutation here, but what happens is it shifts the whole reading of the 3s, okay? And so what's going to be the end result is everything after that deletion or insertion is going to be altered. Okay, so many amino acids can be affected. And that wraps up the lesson on protein synthesis. Okay, there are some videos that you can watch that are very good. You're going to be doing an activity to kind of help review you. In fact, I would like you to start the activity today. If you look at the lab, there's the whole front page, and then it carries over to a picture of a ribosome and like one other question. It would be great if you could finish up the activity to the part where you need the marshmallows and the pipe cleaners. Okay, the actual activity portion we're going to do in class tomorrow. But just to kind of practice, there's some questions about transcription and translation. There's some practicing turning DNA into mRNA and recognizing how the mRNA is complementary to the tRNA. I do want to remind you the only molecule you should ever be using on the codon chart is mRNA. Okay, never use the tRNA or the DNA on the codon chart. But either way, there's some practice for you to do. Then we're going to model this activity in, or model this process in an activity that we're doing tomorrow. So I hope this makes sense. If you have any questions, just let me know. Sorry about the confusion with the first videos. Hopefully this will address any questions that you may have had, if you had any, or if you were absent. Now you can still go back and watch the explanation. But I will see you guys soon, and I hope you have a great day.